All right, so if we are in a eukaryotic cell, we've already gone through transcription in the nucleus, we've already gone through mRNA processing, and then we are ready for translation. And so translation is going to happen out in the cytoplasm. So the first thing the mRNA is going to do is leave the nucleus through a nuclear pore and travel out to the cellular structure that builds proteins. Where are proteins built in a cell? Think back to Unit 1. Yes, ribosomes. So that mRNA is going to go find its way to a ribosome, and then we're ready to translate. Then we're ready to go from nucleic acid to protein. Then we're ready to take what's written in the mRNA and use it to build a chain of amino acids, the building blocks of proteins. Translation is going to require ribosomes and mRNA template. And just like in transcription, there is going to be a certain sequence in the mRNA that means start and a certain sequence that means stop. We are now getting introduced to one of those other types of RNA that I told you about before, rRNA. And in this context, R is going to stand for ribosome. So rRNA is, the ribosome itself is made up of a whole bunch of RNA that's folded all over itself and bound up with proteins. And the ribosome, I like to think about like an Oreo cookie, but just the cookie parts. And so the ribosome is made up of two subunits, a small subunit and a large subunit that kind of come together and sandwich the mRNA in between. So in my analogy, the mRNA would be like the cream of the Oreo cookie. And those two cookies, literally, if we're talking about the ribosome in terms of rRNA, those two cookie parts are made up of all of this sort of interweaved RNA curled up all over itself. And you can see that here. So this large subunit is a whole bunch of RNA. Literally all that is RNA. And this small subunit is a whole bunch of RNA curled up on itself. And these two pieces come and form a cookie around the mRNA sequence that runs through the middle there. Translation is always going to start at a start codon, and it's always going to end at a stop codon. You do not need to memorize these because I'm always going to provide you with a codon chart, so you'll be able to use the code in the mRNA to translate it into proteins using this chart. But if you do want to just sort of be familiar recognizing these, the start codon is always AUG. The start codon is always a sequence of 3 nucleotides, AUG. I like to think, school starts in August, an easy way to remember it. And the stop codon is one of these 3 here, UGA, UAG, or UAA. And if you think back to our PLQ video, remember a codon is just a sequence of 3 nucleotides that codes for 1 amino acid. And this might be a little bit clearer to you when we get to our codon chart in a second. And then our last player entering the scene here is tRNA. So rRNA was ribosomal RNA, mRNA was messenger RNA, tRNA is transfer RNA. tRNA is going to be the molecule that actually adds the amino acids to the polypeptide chain. tRNA is the molecule that carries in the building blocks of proteins and allows the new protein to be constructed by the ribosome. And tRNA is made up of RNA, but it has this really specific little shape here, this kind of clover, I like to think about it. Let me switch my pen color. Let's see, black might be best. So this whole thing here is A's, U's, C's, and G's. And it sort of folds up on itself so that certain sequences, like right here, can complementary base pair to each other and kind of hold together with hydrogen bonds. And then right here, same thing, complementary base pairing. And right here, complementary base pairing. And so you end up creating this kind of little clover shape that sticks to itself, and it has 2 distinct ends. We've got an end up here that carries an amino acid. We've got an end up here that carries a building block of proteins. This is not nucleic acid, this is amino acid. This is not RNA, this is protein. But all of this is RNA. And the special part of this molecule is right down here. The piece that allows this to actually bind to the mRNA is right here. And let me go ahead and erase my markings so we can see this a little bit clearer. So you'll notice at the bottom of this molecule here where we were just looking, there is a anticodon. There is a sequence of A's, U's, C's, and G's that's going to complementary bind to the codon in the mRNA. And so in this example here, if the codon was GCC, the anticodon would be the complementary base pair to that, in this case, CGG. And so we're going to be able to take codon by codon a matching piece of tRNA to come in and carry in an amino acid here that's going to be added to the chain to create a brand new baby protein. And again, if you're thinking this is a little bit too dense for me, let's go back to our drawing and see if we can play with this idea in a little bit more detail. Oh, no, not that. Here we go. Okay, so we're back inside of our cell, and we finished here in transcription, remember, with our mRNA. And in our simplified example here, I'm not going to do mRNA processing because that's just going to overcomplicate it. So let's pretend that already happened. And what's going to happen now is mRNA is going to journey out of the nucleus through a nuclear pore and into the cytoplasm. So I'll just copy over this sequence out here. So we've got 5', AUG, AUC, UCG, UAA, and 3'. And then this mRNA is going to need to find a ribosome. And the ribosome, remember, has 2 subunits. I'll make my ribosome, let's make it blue. And so this bottom half of the Oreo cookie down here, and this top half of the Oreo cookie up here. I'm drawing it as, a simplified version, but this would be, remember, a whole bunch of RNA all folded up on itself, rRNA. So this ribosome is really rRNA, kind of sandwiched. And then this mRNA is going to sneak its way in there and kind of fill the Oreo cookie. Okay, so if we take our sequence here, and let me erase that so we can actually write it out. Let's take AUG. And what's our next one? AUC, UCG, UAA. And it's going to kind of move its way through the ribosome in this direction, so 5' to 3'. And the ribosome is going to read it in sets of 3. The ribosome is going to read it in codons. So it's going to read AUG, and then AUC, and then UCG, and then UAA. And as it does that, it's going to call in the complementary tRNA. And I'm running out of colors here. Let's make tRNA, let's make tRNA white. So remember, it's going to call in that weird little 4-leaf clover-shaped thing, tRNA, that's carrying, let's make proteins. Let's make proteins pink. Okay, that's carrying an amino acid. It's carrying a single building block of a protein. And every single tRNA, this is my tRNA, every single tRNA, whatever its anticodon is right there, is specific to whichever amino acid it is carrying. So the tRNA that matches with AUG is a different tRNA that matches with AUC, and it's a different tRNA that matches with UCG. And whatever it matches with, it carries in a different amino acid. Let's see if I'm fancy enough to pull in a codon chart here. Nope. Bear with me. There we go. Okay, so this is our codon chart here, and you will have been introduced to this in the PLQ. What we're going to be using this for is taking our first letter in the codon. In this example, let's do AUG right there. And so AUG, we want to find it in our codon chart and figure out what amino acid it codes for. And the way the codon chart works is our first letter is down here on the left-hand side, our second letter is up here on top, and then our third letter is over here on the right-hand side. And so we would start with letter A, follow it up to letter U, and then back to G on the right. And so we end up with AUG right here, which codes for MET. And this is an abbreviation for the amino acid methionine. You don't need to know the full names of amino acids, but you should be able to use this chart to figure out which amino acid codes with which codon. And so if we want to go ahead and add our first amino acid to the chain, we're going to start with a MET, because MET is what was carried in by the tRNA that matched with AUG. And so MET is then going to be attached to whichever amino acid is coded for by AUC. And so if we go back to our chart over here, AUC, AUC, there it is right above MET, which is ILE or isoleucine. Again, you don't need to know the full names of these amino acids. And that will be bound to whichever amino acid is coded for by UCG, because that'll be carried in by the tRNA that has an anticodon for UCG. So if we go back to our chart here and we look for UCG, can you see it? Yep, right there. UCG, which codes for serine, SER. And then our last one here, UAA. If we zoom out again, what does UAA code for? Yes, UAA codes for stop. And what stop means is we don't actually add anything else to the chain here. What stop means is that the ribosome itself disassociates. The Oreo cookie comes apart. So as soon as we hit stop, as soon as we hit UAA, the two subunits of the ribosome pull away from each other, and we release this brand new amino acid sequence into the cytoplasm of the cell. My example is super, super short here. My example is only 3 amino acid long. In reality, most proteins are hundreds to thousands of amino acids long. We're going to do a little bit more practice with this in later videos, but that's basically the process of translation. Taking what's written in the mRNA and converting it into a sequence of amino acids, converting it into the building blocks of a protein that are then going to fold into a functional molecule in the cell.